Swayam Prabha. Digital India. Educated India.
Okay, so today we are moving forward from the chromatin to the next stage in the gene expression. So, how that can be regulated. So, we will move to the next step. Okay, next step in gene expression is the nuclear RNA processing. So, this obviously is eukaryote specific, right. So, we are in a developmental biology class. So, we are constantly talking multicellular environment, all right. So, therefore, splicing, nuclear RNA processing or other processing, all of them become important. So, let us look at that. So, if you look at A here, so in both the cell types, so here these are two different cell types. In both cell types you have 5 different messenger RNAs being transcribed, okay, 5 different genes are being transcribed into RNA. And if you look at the cytoplasm, you have C, D, E coming into the cytoplasm in cell type A, but you have A, B, C coming out to the cytoplasm in cell type 2. So, this is nuclear selection in what is getting exported and what is not getting exported. So, even at that level you could have differences. So, there are certain developmental contexts where regulations at these steps become important, okay, in the overall context of the embryonic development. So, you can't always do it at the level of methylating histone or DNA and controlling transcription at that level. So, there may not be enough time for it. And second, these transcripts may be used later in some other stage, okay. So, therefore, they are made in advance. So, that is why these regulations at the post transcriptional step make sense depending on the context, time and spatial context, and one of them is this kind of nuclear selection. And the second regulation at the nuclear level on RNA is the splicing, okay. So, if you look at here, so the central bar diagram tells you the actual gene structure as you see it in the nascent RNA, okay, just transcribed, not spliced. So, you have 1, 2, 3, 4, 5 different exons here, and these vertical lines, these V shaped ones indicate which is going to be joined, okay. So, like for example, this indicates that this exon and this exon is going to be joined and this will not be joined. So, the number 2 will be treated as an intron in that case. So, this splice donor where I am pointing is connected to this splice acceptor here, okay. So, skipping 2 other potential ones in the middle. So, that is one way of splicing. So, you are bringing together exons 1, 3, 5 and in another one you find 1, 2, 4, 5. So, this is alternative splicing, okay. So, alternative splicing is not a minor component in gene expression regulation. So, as you will see in subsequent slides now. So, that is where we are going to spend bulk of today's time. So, this is as a real example for the first thing, that is nuclear selection, okay. So, this is CH in embryo at an early stage you are looking at. This is a phase contrast showing all the cells here. So, here this is an in situ hybridization revealing the localization of a certain messenger RNA which is here, this CY3A. So, that is expressed only in a few ectodermal cells, not all over the embryo, and that is further illustrated in this northern blot, where RNA isolated from ectoderm in lane 1 and RNA isolated from endoderm and mesoderm in lane 2 are run separated, okay, electrophoretically and transferred to membrane and probed with radio labeled probe for CY3A intron specific probe that reveals in this, okay, that is revealed by this larger RNA. And if you look at it, the difference is not much, they are more or less similar level of presence, indicating that in both the germ layer or all the 3 layers, it is being transcribed. It is definitely there in ectoderm and as well as in endoderm and mesoderm. But if you look at the same transcript using an exon specific probe, which is going to hybridize to the large fraction of the RNA there will be mature mRNA present in the cytoplasm, okay, because the processing happens quickly and they come to the cytoplasm. So therefore, it is largely hybridizing to the cytoplasmic mature RNA, and there what you are finding is its level is very high in ectoderm compared to the other 2, indicating that there has been a selection in terms of the RNA in the cytoplasm, okay, what is getting exported. So in ectoderm it gets preferentially exported, but not so in the other 2 germ layers. So now we will continue with this splicing. So splicing, you can have any one of 4 different ways of alternate splicing. So one is in example A, type 2 procollagen, where you are seeing cassette exon. So you have 1, 2, 3 as a cassette and then 1, 2 as another one in 2 different types of cells. Here you have precursor chondrocytes and here you have the mature chondrocytes, okay. And another type would be mutually exclusive. In one tissue type you have one exon but not the other, and in the other tissue type you have the other exon and the other one is not present. So here if red is present, purple is excluded, and if purple is present, red is exclusive. They both are mutually exclusive, okay. So that is one variety. And the third is alternative splicing by splice site variation, okay, 5 prime splice site here, the donor site. If you look at this exon 2, you will see that these lines, particularly you look at the line between exon 2 and 3. In one you have the full exon 2 is included because the splice donor starts after that. In the other one, somewhere at the beginning of this exon itself you have a splice donor, and as a result bulk of exon 2 is excluded when that splice site is used, okay. So you pay attention to these 2 V-shaped lines that tells you the beginning of a intron. So here it begins here, as a result exon is small. So here it begins at the end of it, and therefore here the exon is full. So in this case this has serious consequences. This BCL-XL is actually inhibitory to programmed cell death, okay, it inhibits cell death, and this small one actually induces cell death, okay, it is totally opposite consequences. And in most cancer cell lines this XL version is common. So as a result apoptosis is inhibited, so cell goes it continues to proliferate, okay. So therefore these have serious functional consequences, so that is what I want to point out here. And the other one same thing, but with the 3 prime splicing, okay. So if you pay attention to this first V, the top and bottom, you will realize they end at different places. So these are the 4 mechanisms by which alternative splicing can happen. Does this happen to only a small number of genes or large number of genes? It actually happens to almost all genes. More than 90% of the human genome genes, protein coding genes, are subject to alternative splicing. To give you an idea, the number of genes in C. elegans and human are not very different, okay. But if you look at the organism's complexity, either anatomical or functional, A Homo sapien can learn all of this and come and teach a class, but C. elegans can't do that. Okay. It is a very simple anatomy, having only couple of tubes, but then having all the basic biological process. It has a nervous system, it can sense pain and so on. So, the bulk of the complexity between C. elegans and Homo sapiens come from alternative splicing. Okay. So, therefore, this is a major thing. So, do not take it as an aberration in the gene expression. You will see some examples, really amazing examples. This is one of them, but this is nothing compared to what you are going to see the next one. So, this is tropomyosin, alpha tropomyosin in rat. So, in different muscle cells here up to this, these are muscles, you see different exons included. So, here these colored ones, these green ones are exons present in all the different variants. Okay. And another important thing is sometimes alternative splicing can affect the 3' UTR sequence. These have this 3' UTR sequence, while these have this, and this one has little bit of this and then this. So, the importance of 3' UTR will become clearer when we go to translation regulation, okay, maybe 3-4 slides from now. And here you have some smooth muscle specific exon included in this, and you have striated muscle specific exons, the yellow color ones included only in them. So, by these various permutation combinations of excluding and including exons, you can generate variety of proteins. So, therefore, the definition of one gene one enzyme that became one gene one polypeptide now actually becomes one gene and one family of proteins. So, we are saying family because they do have some similarity, right, some exon sequence is there in all of them. So, that is what it turns out now. And the most complex alternative splicing that we know currently is this Drosophila neuronal mRNA. Okay. This is expressed in neurons, and we will see the functional importance in the next slide, but this illustrates, okay, from one single gene you get 38,016 different mRNAs. That is nearly double that of the entire protein coding genes in Drosophila. Drosophila has only about 14,000 genes. Okay. So, it is nearly double of C. elegans genome itself. So, here you have 24 different exons in this protein, of which the colored ones, if you look at them, any one of these stripes segments can be selected as exon 4. Okay. 12 different alternatives are possible to serve as exon 4, 12 different adjacent sequences. Similarly, exon 6, 48 alternatives are possible, and then exon 9, 33, exon 17, 2. Now, if you work out all the combinations, it works out to be 38,016, and experimental data supports that vast majority of them are all actually produced. So, now let us look at why that is important, how that helps. So, this is more like the VDJ recombination in human B cells, doing a similar purpose here, but for self recognition. So, here is a neuron. Okay. So, here you have branched dendrites, and these dendrites need to know they are coming from the same cell body. Okay. So, they are essentially sisters, and there is no point in they connecting to each other, they are not going to get any new information. They need to connect to adjacent neuron or another neuron in that chain, then only it will be useful for nerve impulse conduction. Okay. So, this self repulsion is provided by that unique discam protein alternative spliced version produced in that neuron, and another neuron has different alternative splicing, therefore, its own pin code, okay, in terms of discam exon composition, and that is what helps this avoidance shown in B and connections sorry, here avoidance is not shown. So, this is one connection, this is another connection, green and purple, and here green and red. Suppose let us say, I mutate this, discam is not produced. So, what would you expect? These will not know to repel and they will all get adhered and they will make a mass of connected neuronal structure, and that is what happens. Okay. So, this is wild type, highly branched, here joint. Okay. It is all experimental data. So, this is real life, we are dealing with life and biology. The two things we understand here, one is there is whole lot of new mechanisms out there about which we have no clue at all, and we need to discover them, we have to go and discover them. And alternative splicing is one of them, and we are going to see next example where you will find only couple of decades ago we recognized the existence of that phenomenon. There is no way to predict that it is out there, you need to go there and see it, and you need to see the functional consequence in a mutant like this, shown in D here, neuron lacking discam. Okay. So, this is alternative splicing. And another example, this is an interesting example because this work happened exactly around the time I joined the department as a postdoc, and this paper had come out from another lab in the same building, and it was in the press and it was talked about, and I was excited to read, and that is there in the textbook. So, we will read about this. So, here you have a muscle specific splicing issue. So, you have mutations now. Okay. Now, we are going to look at disease conditions. So, in Drosophila visa we mutate and find the function. So, in human patients is there a consequence when these splicings do not happen as they are supposed to be happening. So, we will see an example here. One example here what we are seeing is a muscle specific situation. So, here if you look at the top horizontal bar, in the intron between exon 1 and 2 you have a premature stop codon. I should not say premature stop codon, there is a stop codon. If you translate from exon 1 in the same reading frame into this intron, okay, that will happen if the splicing failed and this intron sequence is included, and that happens when you have a mutation in the intron. Okay. And due to that, a G in the intron, okay, it is not affecting any amino acid sequence, and the G becomes a, and due to that, that promotes splicing starting from that point, okay, around that area. So, therefore, a part of this intron is included, and due to that you get termination and you do not make a functional protein. Okay. So, in this case that stop codon would never be encountered because the whole thing is excluded, and as a result you straight go to exon 2 and that the reading frame continues, and here you make this protein. So, this name of this protein is myostatin. So, this one prevents muscle cell proliferation at appropriate time and they differentiate into muscles right away. So, you do not make too much of muscle, you make less muscle, okay, normal required amount. So, in a family where they found this, 4 guys were already athletes and a small kid, I forgot the age, but it is a elementary school child or even less than that, was able to hold a 3 kg dumbbell in both hands with both hands fully extended, okay, because its muscle was so strong at that age. And we can do it in model organisms, and this is here, this is the mighty mouse, okay, this normal mouse, this is the mighty mouse. So, that is why it attracted press. The paper came in Nature, and then this faculty was on press meet with TV, radio and newspapers. So, you see the muscle difference, right, between the two, because the cell proliferation was not stopped at the right time. So, they made lot more cells, and when they differentiated they made lot of muscles. So, will a developmental biologist right away recommend to a physician that you mutate and make more muscles? No. Will not, because developmental biologists will think what will be the other consequences, right. For example, you will worry about methylation patterns when you think of only the coding sequence. So, similarly you will expect there will be other complexities. You will think there will be a phylogenetic and ontogenic reason for why that cell division stops at that. So, you will not immediately mess up. So, but this gives you an idea of what happens when you have splicing affected. Alright, I think that conveyed the message about the importance of splicing. So, next we will move on to translation regulation. So, translation regulation is very key in certain contexts. Okay, two examples are really outstanding examples to illustrate this point. One is early embryogenesis and another one is the adult nervous system. Okay. You take a neuron for example, it has a cell body and has a long axon, and at the end of the axon you have synapses, and you have to at the synapses at the synapse what do you have? You have to produce neurotransmitters and secrete, and then the postsynaptic neuron must have the receptor to accept it, and if these things have to be produced and degraded very quickly and very rapidly to sync with the nerve impulse conduction, you can't go ahead and unwind the DNA by modifying the chromatin and then transcribe and then get permission from the nucleus to come out, and before that splicing all that. You will not have time for it, and after that the protein has to be transported to the end of the axon to get to the site of synapses. So, that problem is usually solved by making the RNA and keeping it stored where you want, and when the correct signal comes you translate. Okay, that is one context. The other context is you think of the cleavage in the embryo, right, or it is rapid and quickly happens, and during all that time you are not going to be able to do the new transcription to take care of the embryonic development, particularly coordinating the cell cycle and producing lot of cell membrane, right. You are making lot of new cells, so cell membrane, and in each one of them cytoskeletal elements you need to make all of that, and lot of chromatin you need to make. You are increasing the number of copies of genome, right, from just 2 copies in the first nucleus. Now you are going to make lot of nuclei and you need that much of chromosome. You need to make all of them, and then the cell division cycle need to be regulated, and then once you finish cleavage, and in some organisms even during that time cell fate specification happens, like for example in C. elegans at the first division itself the fate of the 2 daughter cells are different. So, fate specification also need to happen. So, all of that you can't do by controlling transcription. So, there again translation regulation of mRNA produced by the mother and deposited in the oocyte plays a critical role. So much so that in some organisms you can get rid of the nuclear components, but still the cell cycle can actually happen without the the nuclear material, because cytoplasm has the required things to simulate that, and these are contexts where translation becomes important. So, now let us see how the translation regulation happens. Okay. So, one mechanism by which translation regulation can happen is increasing or decreasing the stability of the messenger RNA, meaning once transcribed how long will that messenger RNA survive before getting degraded. Okay. So, you can have a half life for that, the duration taken for the messenger, half of the messenger RNA to be degraded. Okay. So, if you take in a lactating mother, the prolactin mRNA normally produced in the mammary gland gets quickly degraded. Okay, percentage of so this experiment I should explain the method first. So this is mammary gland cells, I guess mouse mammary gland cells cultured, and you incubate the cells with radioactive nucleotide, like in this case probably UTP. So all the RNA synthesized gets radioactively labeled. Then wash the cells of that medium containing radioactive nucleotide and put in fresh medium without radioactive nucleotide, but you have unlabeled one. So that is called the chase period. Now you watch the labeled RNA how long it stays. Okay. So without this hormone, the lactation stimulating hormone prolactin, it gets rapidly degraded. Half life is probably about 2 hours or 1.5 hours. But with the hormone it stays for more than 2 days. So this is by increasing the stability, then the mRNA is available for multiple rounds of translation. So you increase the protein product. So the ultimate goal of differential gene expression is to vary the kind and amount of proteins from one cell type to another cell type. So that is the ultimate end point of gene expression when you talk about protein coding genes. So this is stability, an example for differential stability. So here the stability is under 2 different conditions. The next one that we see is what normally happens in oocytes. This was first described in good detail in Xenopus oocytes, so therefore we are going to look at that example, and this is the kind of regulation that is critical in almost all the oocyte development that people study. Okay, so in fact in our lab it's exclusively we study translation regulation in the germline. So let us look at this. So here okay, before that I should introduce you this idea of looking at the mRNA. So normally you write mRNA as a long line 5 prime cap to 3 prime poly A tail. But in reality in cells they do not exist like that, they instead are found in ring like form. Okay. So the 5 prime end and 3 prime end are brought together by proteins that bind to the 5 prime cap, like for example translation initiation factors that bind there to the 5 prime cap and 5 prime UTR, etc. They interact with proteins that bind to the 3 prime UTR or 3 poly A tail, and those protein interactions make them into a circular structure. Okay. So that is what you are seeing in this electron micrograph, and here is one example. So normally the initiation factors binding to the 5 prime cap, they interact with a protein that binds to the poly A tail, okay, called poly A binding protein, and that is how this circularization happens, and that is required for binding of other initiation factor like 4G, which recruits the small ribosomal subunit and initiate translation. Okay, so that is what normally happens. But in Xenopus oocyte, the most mRNA that are not translated at a given time, they are found in circular form, but not by the interaction of 4E 4G with the PABP. Instead a protein called maskin binds to 4E in preference to binding 4G to 4E, and that maskin interact with a protein called cytoplasmic polyadenylation element binding protein, okay, CPEB, that binds to this polyadenylation signal sequence, and that interacts with maskin, which interacts with 4E, and this kind of circular form totally exclude the other initiation factors, and therefore it is translationally dormant, it does not get translated. And when you have appropriate signaling, for example progesterone hormone, which stimulates oocyte maturation and complete the meiotic division upon fertilization, and that leads to an activation of a kinase that phosphorylates the CPEB. Okay. And when the CPEB is phosphorylated, it recruits a protein called cleavage and polyadenylation specific factor, and that recruits a poly A polymerase, that is gold 2, okay, for those who know C. elegans lingo. So, it recruits that poly A polymerase which extends the poly A tail in the cytoplasm. Remember when we learnt about eukaryotic gene structure, I said some polyadenylation happens in the nucleus, and in some of the developmentally regulated ones additional polyadenylation happens in the cytoplasm. So, this is an example of that. So, you have further extension of the poly A tail, and this poly A tail now binds the PABP, okay, and that can now interact with 4G, and this phosphorylation also throws the masking out of interaction with the 4E, it gets displaced, now translation starts. So, sequentially the mRNA, that has not yet been totally worked out how this is choreographed. This gives you a mechanism of how activation happens, but then there is a wave of sequential activation. Some mRNA are expressed at certain stage, some mRNA are expressed later, and so on. So, that is still being intensely studied. So, this whole mechanism was worked out at the beginning of 2000, like the papers were coming out in 2001, 2002, around that time. So, now this mechanism has been found in many other organisms where you can do a lot of genetics. So, they have done a whole lot more work on many examples of this kind, okay. So, let us look at one such example. So, that is the Drosophila oocyte. So, where you have a protein called a bicoid. So, this bicoid is actually an interesting protein, it is a transcription regulator as well as a translation regulator. Here we are seeing its translation version of it. So, it binds to a sequence called bicoid recognition element on an mRNA that encodes a protein called caudal. So, this caudal is required for posterior development, or rather to prevent the posterior development in the anterior, okay. So, this needs to be suppressed, and that is done by bicoid, which promotes normally anterior development, and when it binds to this, it recruits this another new protein which binds to the methyl cap, and this excludes all the initiation factors. So, this is how it prevents the translation of caudal mRNA, okay. So, now you realize that the general theme here is proteins that interact with RNA sequence can interfere with translational activation, okay, and some of them actually increase or decrease the poly A tail in some contexts, not in Xenopus oocyte context, where even with shorter poly A tail the mRNA are stable, it is just that they can't bind PABP and therefore they are not translated, okay, but they are not degraded. In certain cases, if you remove poly A tail, the RNA is marked for degradation. So, here there are some examples of the molecules, mRNA molecules that are regulated and where they function and in what organisms they come from. These are listed here, the list continues, okay. Cyclin, not surprising, right, cell cycle regulation. So, it goes on, okay. So, from multiple organisms. So, this is obviously an evolutionarily well conserved mechanism. Alright, next one kind of obvious, but then it took a long time for people to find an example of this. If proteins can bind sequence specifically to 3' UTR and 5' UTR, why not another nucleic acid, right? Sequence complementarity should give you a lot more specificity to the given sequence, and this was found in an interesting screen in C. elegans embryogenesis. So, in C. elegans embryogenesis you have a certain pattern of division that can actually end up reiterating if certain gene expression changes do not happen, okay. Let us say at 4th cell division level a certain pattern of asymmetry is generated during cell division, and you do not want that to be reiterated again, because you will end up producing similar kind of cell fates in another generation as well, and that is avoided by blocking some gene expression. So, mutations in them are called heterochronic because in the time order they are at the wrong time. Okay. So, what should have been at the third stage will end up repeating at the fourth stage or fifth stage or even later, and therefore they are called heterochronic mutations. And one lab that identified a mutation that had a heterochronic phenotype, when they finally mapped, they did not find any proper protein coding sequence there. And when they they were very certain because the phenotype is clearly there, and their mapping they were very confident about the locus they mapped, and they knew that that locus matters even if it is not encoding protein. Then they carefully looked at and then they found it encodes short RNA, a small RNA sequence that is complementary to the 3' UTR of another RNA that is involved in this lineage specification. So, lin stands for lineage. Okay. In C. elegans most genes have 3 alphabets and a hyphen and a number. The number usually is the order in which that particular gene belonging to that class was identified. So, lin meaning lineage defective, in that it is the 14th gene that they identified. So, if you look at lin 14 mRNA sequence, in the 3' UTR there are the colored sections, okay, 1 to 7, and those are the sequences to which this lin 4, okay, that is one of the heterochronic mutation which was not encoding a protein, and they found it is a small RNA, and that is complementary in this fashion. The loops indicate where there is no complementary, okay, where you see linear thing, there is base matching. Okay. So, this was the first discovery, and is this something nematode specific and you are not going to see? That is not true. Now, in the last 20 years people have discovered these small RNA are naturally encoded in the genome just like the C. elegans genome, and they regulate the translation of vast majority of mRNAs. The current estimate is about 30 to 40% of all human mRNA are subject to regulation by these small mRNAs called miRNAs, okay, micro interfering RNA molecules or microRNAs shortly. And this is how they are made. They usually exist in multiple tandem repeat copies, and they are transcribed, and once transcribed you have a nuclease, a drosha that cleaves. So, the information in this cartoon comes from studies in multiple organisms. Okay. The first example is from C. elegans, that is where it was first discovered, but then people worked out the mechanism by studying multiple organisms. For example, drosha was discovered in Drosophila, where they made extracts, cell extracts, and did these reactions in vitro. So, these multiple repeats are digested into individual units in the nucleus by drosha, and that is transported into cytoplasm, and see these come as hairpin repeats, okay, and that hairpin is removed by a cytoplasmic nucleus called dicer, and now you get the 2 strands and they are unwound and loaded on a complex called RISC. Okay, this RISC complex is the one that takes the one of the 2 strands of the miRNA duplex and uses that to identify the target sequence and goes there and binds to that, and binding to this can have multiple consequences. One of them is the RNA gets cleaved, okay, the stability is reduced dramatically. This is one, but you can have multiple consequences that is there in this, then we will go back to that example. So, this miRNP ribonucleoprotein complex carrying that sequence, miRNA sequence, can do any of these 3, like for example, it could inhibit initiation complex, a protein binding to the 5' cap, or it can inhibit ribosome's ability to elongate, it can interfere with that process, or it can remove the poly A tail and reduce the stability of the RNA, or it could actually recruit proteases to digest the nascent peptide that is coming out. All 3 mechanisms have been seen with specific examples, okay. And does that to what extent it matters, like in a mammalian context. So here you have the lymphocytes context, where development of the different kinds of lymphocytes, and if you see, if you have a lymphoid precursor cell which is usually low in this miRNA, usually called MIR and some number, so MIR 181, so you get both B cells and T cells, and B cells express MIR 181 in larger abundance than the precursor. Okay. But if you artificially introduce large quantity of MIR 181 in this precursor cell, it will end up producing among the daughter cells, vast majority will be the B cell type and not the T cells. So you make more B cells at the expense of T cells. So these have clear developmental consequences in multiple organisms. Okay. Well, that is what the fate is, in the MIR, whatever it is going to prevent expression, preferentially leads to B cell formation and T cell differentiation does not happen. So each cell type is going to have one specific set of gene product, not genetic information, gene product, and as a result its fate is going to be different.